Is my screen nice? Okay, we're recording this, so hopefully I'll upload. Did you guys get the slides? I don't know if you heard her say it, but she just emailed you guys all the slides. If you want to like follow along, right in your iPad, whatever. Okay, we'll get started. Okay, there is 101 slides, but however, most of those are just questions with like the question, and then it's like duplicated with the answer, then does that a bunch. So like it's not actually like 101 separate slides. So please don't freak out. We designed this so that hopefully most of today is like questions instead of just like trying to reteach you everything. Like we'll still go over everything, but we want you guys to get some like actual practice in. We have some of the quiz questions that we've had up on here. We have some of the problem set ones, and just like some random ones. So should be good. And I'm Angelique.
And I'm Jenna.
So, okay, three topics on the exam, not four like the first one, just three. So it's cell signaling, microtubule stuff, cell cycle. If you just came in, we did like a little hands poll. We were like, what's kind of like the worst one that you guys need the most help with on here? Most people said 5. Are we still in agreement on 5? Still in, okay. So we'll spend most of our time on that. Honestly, I think that's where most of our slides are.
Yeah.
About to. So, okay. So we're going to go through the basic stuff a little quick because I do want to get to the questions. But if anyone has any question, anytime, raise your hand. Jenna can answer. I can answer. Like, don't have to wait for me to finish talking. But we are going to go through like the first part of this a little fast just so we can like actually get to the big stuff. Okay, so principles of cell signaling. Somebody give me a reason why a cell might receive a signal. Live, survival. What else? Yeah, differentiation, dividing, right? Stuff like that. So different signals do different things. Cells might need signals for different things. Pretty straightforward. I'm just going to go straight to this question. This was in the topic 5 review. A lot of these are still from the topic 5 review that we already had, but with a bunch of new questions. But this is still important. So somebody, if a signal, let's do the first one first. So if a signal can pass through the phospholipid bilayer, it is blank and blank. Anybody want to tell me?
Smaller and hydrophobic.
Smaller and hydrophobic. Okay. So if it can't, then it has to be what? Larger and right. Okay, let's get into why. So smaller, let's go back to this like picture real quick. Okay. It'll come back in a second. Okay. Bilayer, right? What's in the middle here? Lipids. Lipids are fats, right? So the tails here are hydrophobic, heads hydrophilic because it's in the aqueous solution, wants to touch water, right? So if it can pass through, it has to be small to kind of get through here. And it has to be what to get through this middle part? Right. So if the middle is hydrophobic, because lipids are fats, they're hydrophobic, it needs to get through that part. Pretty straightforward. If it can't pass through, it's large enough where it can't go through. And then if it's hydrophilic, then how the heck is it supposed to get through the middle part, which is all hydrophobic? Make sense? Cool. So 2 outcomes of cell signaling. There is one, which is altering protein function. So protein's already made. We're just going to change what it does. So if you guys remember the term conformational switch, so like adding a phosphate to something to turn on, taking it off, that's a switch, right? You're turning something on and off. That's just altering its function, right? There's also altering protein synthesis. Synthesis means we're going to make it from scratch. We're going to remake it to do something else, or we're just going to make a bunch of stuff so we can get a certain thing going. So these 2 things are not mutually exclusive. So they can both. Okay, let me back up a bit. Okay, so different cell types can express the same receptor. So 2 different cells, same receptor, and you can get different outcomes. So quick example, acetylcholine in a heart muscle cell, it's going to lead to decreased heart rate, force of contraction. In a different type of cell, it's going to have secretion of saliva, right? So it can be the same signal, but depending on the type of cell, can have a different outcome. So this is also from the topic 5 review, if you guys remember, but still super important. She kind of hammers this a lot in your face, but let's just talk about it real quick. So we have the two outcomes, which is altering protein function, and then there's altering protein synthesis, the making of stuff, right? Why might altering protein function be a faster way of making an outcome occur? We want something to happen. Why might altering the function of that protein be faster than altering the synthesis of that protein? Somebody. I already know you know it. You were at mine. Yep. Right, exactly. So things like transcription, translation, those things take time, right? We don't got time. You could still do it. It could still work, right? It's still a solution. But if we have a protein that's already existing and we just change what that guy does through whatever method we want to, it's going to be a lot quicker to do that than trying to go from scratch, go through transcription, translation, all that good stuff, getting it through the system to make something happen. Make sense? Okay. It'll come back in a second. Okay. Signal amplification, crosstalk. So signal amplification, it's like gossip. So if I tell Jenna something, Jenna goes and tells 2 people, each of them go tell 2 people, a message is spreading. That's literally what signal amplification is. So in this case, we're going to go over some different ways of like how to get a message across faster, but that is all what signal amplification is we have a message, we want to get that message somewhere. Do we want to get it slower? Do we want to get it faster? Who do we want to get it to? That's just signal amplification. So I have a message and I want to get it out. How do I do that? So crosstalk. Crosstalk is different pathways, different linear step pathways talking to each other. Does anyone have a guess as why this would be helpful? Why do we care about crosstalk? What's the purpose of crosstalk? Anyone? Any guess? Why would I want to talk to another pathway?
Pathway could be like not functioning, but you want then so different pathway can then like cause that to be active and that in some cases that can happen.
Yeah, so like that's a good example. So one pathway, maybe one pathway's off, the other pathway is like, nope, I want you to be on, turn something on in that pathway, and then we get the outcome. So one protein from one pathway may talk to another one from another pathway, interacting and changing the function of that other protein in some way. It just means when pathways talk to each other. And the reason for this is so they can coordinate their responses. So if you guys remember from Unit 5, like TK1, like that whole thing, there is always the possibility of crosstalk. So even if I don't get the signal, if another pathway activates something downstream in that pathway to get the outcome, I can still get the outcome. So if she ever asks you a question that's like, oh, like specifically regarding cell signaling, and she's like, oh, well, if this is missing, can the outcome still happen? The answer is no, but there is always the possibility of crosstalk. It's never in bio, never say never. Like there's always something going on. There's always like exceptions, and crosstalk is always a thing.
You have to bring that up
In your response, I don't know if she is going to ask you something like that straight up about cell signaling, but I would mention it. But I would always say like no first. Like if we're missing that, no, assuming no. But crosstalk is always a possibility. It never hurts to mention that. So someone give me some pros of having more steps in a pathway versus less steps. Somebody give me a pro. More opportunity for crosstalk. Perfect. What else? Yeah, perfect. So more amplification, right? So if I have a message I want to get across, there's more steps. That's more people talking to other people. Does that make sense? So those are the pros. That's why we would want to have more steps. So again, more amplification, more steps means there's more opportunities to talk to each other. And then there's crosstalk. So each step, there is a way for a crosstalk to come over and say, hey, I want to turn that on. Or hey, I want to turn that off. There's more opportunities to do so. Make sense? Okay, cons. Give me some cons. Why would we not want more? More time consuming. Yeah, it's just going to take more time. There's more people to tell. It's going to take more time. Maybe when we want to do something really fast, and that's not fun. Does anyone know what the term, I think it's like rate limiting diffusion kinetics. Have you guys heard of that term before, kind of? It's basically, it's the likelihood of two specific molecules bumping into each other when they're just floating around. That's what that fancy term means. So if we have more steps, then we have more diffusion rate limiting kinetics, and so they're slower, can't get it out. It's pretty straightforward. Okay, pathway. Okay, don't be scared. Don't be scared. A lot of these pathways are simpler than you think. There's a lot of random terms that you'll have to remember, but the actual act of it is pretty straightforward. So I would say this is like the easiest to remember for now, and then we'll get into the GCPR stuff, which I know a lot of people have questions about, and the JAK-STAT one. But let's just go over this again super quick because I want to get into the questions. So first, can somebody tell me what a dimer is? What does it mean when there's a dimer? Yeah, two things come together. So when she says dimerize or dimer, I think she says that about like the kinesins. Dimer is just when two things come together. Okay. So first thing that happens, signal binds to receptor domain outside the cell, which causes a conformational change. What do we mean when we say conformational change? What am I changing? Shape, right? Changing the shape. So you change the chemistry of something, that changes the function. Do you guys remember the structure determines function thing? Still here, okay? So anytime structure is changed, function is changed, okay? So conformational chains, two RTKs come together. What is that called? Dimerization, right? Activates them as kinases, comes together to form the unit, and then they phosphorylate each other. So that's autophosphorylation or cross-phosphorylation. It's the same thing. They're just phosphorylating each other. The phosphorylated tyrosine kinases serve as the binding sites for downstream proteins called the signal transduction cascade pathway. So that's all the TK1, TK2, but you don't have to know the specifics for that one. Let's talk about RAS. So the adapter protein, aka green guy, call it green guy, GRB2, binds to one of these phosphorylated tyrosine kinases on the RTK through the SH2 domain. We're going to get into like the difference between those two. But remember, the SH2 domain binds to what? It'll come back. Phosphorylated tyrosine kinases, right? That's what it binds to. So GRB2 binds RAS-GEF, which is the SOS protein. It binds it through the SH3 domain with the PXXP motif. So the GRB2 has the SH2 domain, the SOS has the PXP motif, and they bind to each other. Okay? That RAS-GEF SOS will change RAS from GDP to GTP. What is the difference between these two? Does anyone remember GDP and GTP? What's the difference? Yep. So one I kind of interpret as like being on versus one being off. Right. GDP has 2, diphosphate. GTP has 3. We'll get into like the little trick that we kind of use with that in a bit. But RAS-GEF is going to change RAS from GDP to GTP. We're also going to get into what GEF is, what GAP is, in case you don't remember any of that. We have all that here. So, little trick. You said it earlier. When RAS is in a GDP bound state, it's what? On or off? Off. When RAS is in a GTP bound state, it's on. Okay. So, little trick, general rule of thumb. When you see GTP, think on, think active. When you see GDP, think off. Okay. Just kind of a rule of thumb just for this unit. It doesn't apply to everything, but in everything that we talk about, that's pretty much the case. So, RAS GTP, which is inactive or active now? Active, causes the activation of MAPK3. We don't really know how it happens. It just happens. And it causes the MAP kinase cascade, which just means K3 activates K2, activates K. Like just remember 3, 2, 1. Just remember the term MAP kinase cascade. And yeah. Oh yeah, sorry. It switched. Yeah, I'll fix that in the slides. But GDP off, GTP on. Okay. Thanks for pointing that out. I'll change it. Yeah, map cascade, kinase cascade, 3, 2, 1, map K3 to map K2 to map K. Cool. Okay. So survival signaling. Why do we need survival signaling? What is going to happen if we don't have a survival signal? What's the term? Apoptosis, meaning cell death. So it needs constant survival signaling, otherwise it's going to die. Pretty straightforward. So the signal binds to the RTK, this first survival signaling. Receptors dimerize, which means what? Two things come together, phosphorylate each other. PI3, this guy. So now these two have come together, and now it's going to bind the phosphorylated RTK. So signal comes in together. Now they can activate each other, and now PI3 can come in and bind to that phosphorylated RTK through the SH2 domain, because what does the SH2 domain have? What is it? Phosphotyrosine kinase, that term, that is SH2, always remember. So now, this is activated because now it's binding to the RTK. And now it can phosphorylate what we call PI, this long term here. I never memorized it, I just thought of it as PI. Phosphorylates this, what we do when we phosphorylate something? Are we adding or taking away? Adding a phosphate. So it adds it to PI, we get PIP, which she calls PIP3. They're the exact same thing. So this is from this. Now we add a phosphate, this. So this is from the same photo, but it's just the second half. So the PIP, which is now activated, this guy, can now serve as the binding site for this brown guy, PK1. Okay? AKT is the central regulator. So this guy is kind of what depends on everything, pretty much. She mentions it a lot. We'll talk about some questions with this. Green guy is AKT, okay? So PK1 binds to AKT. And then PK1, sorry, and AKT also binds PIP, which is that third guy, this guy. Okay? This is the same thing. So once this happens, once they all bind, it phosphorylates AKT and causes AKT to release from PIP, okay? And diffuse freely in the cytosol, but it's not fully active yet, okay? Please remember this. So when AKT changes conformation the first time, it's not fully active. It's not ready to go and do its thing. When it actually can go and do its thing is when the purple guy, so PK2, goes in and phosphorylates AKT, okay? So it needs two activations. The first one is by who? PK1 and PIP, right? So AKT binds blue guy with the 3. PK1 also binds. And this is what causes the first activation. Make sense? Yeah.
Okay.
So, I think it's 2 different PIPs. Yes.
It is 2 different
It is.
PIPs.
But they're both still, they're both needed. So PIP binds to protein kinase 1 and PIP is also with the AKT.
So does it matter if we know like when each of them bind to the PIPs? Like both of them just bind to their separate PIPs and then
I would say when you're talking about like activation of AKT, if she asks you anything, what's key is the PK2 coming in. So she's probably not going to go into detail about like those two separately being binded. You're going to know that PK1 is going to bind at some point. You're going to know that PK2 is going to bind at some point. PK2 is when that full activation happens. Okay. So first one is by PK1. Second is PK2, and they're both attached to that PIP. So when that happens, now it can phosphorylate bad. Okay. And this is, I used to get really confused with this because it's the cell death inhibitory. So then I have to like work backwards in my head, but don't worry. You're just remember that AKT phosphorylates bad. Okay. So when it's inactive, bad and BCL2 are together. Okay. When it's phosphorylated, when bad is phosphorylated, it lets go of BCL2. And when that happens, now it's going to prevent cell death. Now it can survive. If they stay together, what's going to happen? Cell dies. So that's kind of how I remembered it in my head. If they're together, bad and BCL2, cell dies. If they're apart, cell's going to survive. Okay? So try not to get too mixed up in your head with it like I did. But I kind of like that little trick. Just if they're together, it's going to die because nothing's stopping it from dying because the cell signal hasn't gone all the way through. And then if it gets phosphorylated, it's going to let go of it. It'll survive.
Is it similar, the relationship between bad and BCL2 similar to E2F?
Yeah. Yeah, I was actually just about to say that. So it's just like RB and E2F, right? Like going to the restriction point, when it lets go, it can like actually go and do things. So yeah, it's pretty similar. Okay, flow process, again, kind of speeding through this because I want to get to questions. So this was on your problem set. This was the one that was graded for accuracy. Okay, we're going to go to this one, and then we're going to do the next one. That was also graded for accuracy. So this is going to be one I want you guys to like at your tables or by yourself, think of some. Let's just start with the gain of functions. Come up with some ideas for what gain of functions would lead to the survival pathway always being on. If it's always being on, is bad and BCL2 going to be together, or is it going to be apart always? You're going to be apart always, right? It's always going to survive. So that's just all it's asking. So like in cancer and stuff, you can't kill the cells that much because they have mutations. So they keep differentiating, they keep spreading, they're alive. That's kind of this question. Whenever she says cancer, it's like, it just means that like you can't stop the cells from dying. So let's do it at your own table for like 1 minute. We'll come back, we'll go through some gain of functions, and then we'll talk again about some loss of function ideas, and then we'll go over it. You're totally getting Yago would. I would like be on his phone. Yeah, you could totally sit. Just sit here, like at the same time. You could sit there. If you want to be on your laptop and you don't like it having on your laptop, you could always go to. I don't think anyone there.
So I can do microtubules and then
We could just go back and forth, I guess, for cell cycle, but to be honest, I'm not the best for cell cycle.
I can do something cool and then I can do it.
I think microtubule is like a little bit more straightforward. So that's something that we could like do to put together. I don't know. I can just like fill in whenever you're like tired.
You're good at
No, you know why? Because I was this stupid kid, and I would always be off thirds. So I like don't get it. Like I didn't get it until I made that presentation. Like at all, like for topic 5. Like I didn't get it. I don't know how I became a teacher. A lot. So a lot. Okay. All right, class. Okay. Somebody give me an idea for gain of function. Give me one example. I know you got it. Just want to give somebody anything. AKT, who said that? Who said it? Why? Oh, it's phosphorylating bad, which means bad's gonna let go of what? Which means it's always gonna be on. Next, I like that one. Yes. RTK has a gain of function, so it doesn't, it's always on, doesn't need the signal. Yeah, I like that one. Next. Come on. There's more.
I guess the loss of function is bad because that's fine BCL2.
Yeah, you have a loss in function in bad. Yeah, I like it. Okay, I'll show you guys the gains right now, and then we'll just get into the losses real quick. But so gain in function in RTK. Good answer. Gain in function in AKT. So what does that do? Activated AKT can go and phosphorylate what? Bad. Bad lets go of, which means cell survives or dies. Survives, right? Always on. Okay, gain of function in BCL2. Why would this be a way to keep the survival signaling always on?
And that would be.
Yeah, I think the implication here is that like if it has a gain of function, then it can like it's activated, right? So it can just like release from bad, which means that it would be on. But like yeah, like the survival signal is going to go through. Okay, gain of function in PI3 kinase. Why would this be correct? So when she says PI3, it's the what PIP3. That's what she's talking about. Why would this be helpful? What's it gonna do? What's it gonna do? What's gonna do? Yeah, you guys want to answer? Come on. Come on. What does PIP do? What does it do? Right, let's go back. Yes, yeah, kind of, right? Yep, it halfway activates AKT, which lets it go on to the next activation, right? So it's fully activated. So that's another answer. But honestly, I like the first three the best. I feel like they're the most obvious. Okay, and then we had the loss of function, right? So he said, okay, what if there was a loss of function in bad? Bad can't release BLC2. What's the cell gonna do? Sorry, I mean, gain loss of function in bad would mean that it can't hold on to BCL2. That's sorry, that's what that means. Okay, does anyone know what P10 is? Anyone know? We'll get into it, but that's loss of function P10. We'll get into it. So, more fun problem set questions that were great on accuracy. So, there's a phosphatase called P10 that takes off the phosphate, right? Phosphatases take off. What adds phosphates to stuff? Kinases. Takes off the phosphate that PI3 kinase puts on the whatever, that's just PI, right? That's just this guy. If you guys can't see, that's PI is just this guy, right? So there's a phosphate that takes off. Would a loss of function in this phosphatase cause cells to undergo programmed cell death and or prevent programmed cell death from occurring? I want you guys to talk about this at your table for like 30 seconds and then we'll come back. Go ahead. Okay, let's get some answers going. Let's get some guesses. I really don't care if you get this wrong. I'm not even 100% sure if I'm right in my head. Yeah. Silence.
I feel like it would just prevent programmed cell death from occurring like normal because normally the phosphate isn't taken off, it's just put on. So if there was a loss of function in the taking off of the third phosphate, then it would just be normal.
It would just be normal. What do you mean?
Like it just occurs like it normally does.
Yeah, it's just gonna keep going, right? Anyway, yeah.
To continue to keep programmed cell death.
Yeah, right. Okay. So yeah. So okay, let's go through this, right? P10 takes off the phosphate from this guy. So when he has 3, can go and do its thing, bound to PK1, bound to AKT, he's ready to go. Why would we need a phosphate, like a phosphatase, to take off that guy? Why would we need to do that? Why would we need to have it go back to normal? Just to reset it, right? So normally, this is apart from this question, normally gets phosphorylated, goes and does its thing, and then P10 comes in, takes it off for it to go back to normal, right? We don't always want the cell to survive. Sometimes we want the cell to die, okay? So if P10 can no longer take that phosphate off, is the pathway always going to be on or is it always going to be off? So it's always active, right? So P10 comes in, takes off that third phosphate to reset it back to normal. So we've done it. Okay, we've sent our survival signal. We're done with this now. Let's take off. So if that can't happen, if he can't take it off and it's always on, then the pathway is always going to be on. Does that make sense? P10, phosphatase. Any questions? Okay, yeah, this is what she says in the answer key. Did she send the answer keys out to you guys for anything? Everything except 7? Oh, okay. Yeah, so she sends out answer keys. Also, if you guys aren't using the topic notes yet, use the topic notes. When I took this class and I didn't understand the way she said something, like topic notes were so goated. Please use those. So,
And watch the lecture videos that
Yeah.
she sends out.
yeah, guys, like please
Please watch the videos.
please, like please help us out. Like please watch them. Okay, yeah. So quick recap. So P10 dephosphorylates PIP to PI2, resetting it, right? So signaling can't happen. Signal can't get across. So at the end of the day, this would always be on, meaning the cell would always survive. Okay. Somebody tell me what would happen if AKT had a loss of function. So remember, AKT is green guy. Green guy. So what if it had a loss of function? What would happen?
The cell would go into program cell death because the CL2 wouldn't like come off of bad. It wouldn't bad wouldn't be phosphorylated. So
Yeah, no, that's a perfect way of putting it. So if AKT can't phosphorylate bad, bad is going to stay holding on to BCL2. And if they're together, if they're not letting go of each other, is the cell going to live or die? It's going to die, right? Yeah, exactly. So AKT when active, phosphorylates bad, causes the cell to live. If that's another way you want to think about it. Phosphorylate bad equals live, unless there's something wrong with BCL2, but normally, right? If AKT can no longer prevent apoptosis, the cell will die. So if it can no longer do its job to phosphorylate bad, bad's going to stay onto BCL2, cell's going to die. Okay, SH2 versus SH3. Quick recap. What is SH2? What did they bind to? They're what? They're what? They're this. Phosphotyrosine binding domains. Okay. Sorry, I think that's supposed to say SOS. I don't know why it just says SO. So examples of it, right? SOS. Sorry, SOS is the other one, I think. SH2 binds to phosphorylated tyrosines on other proteins. GRAB2, remember GRAB2? Where's GRAB? Where's that guy? GRAB2. Yeah, GRAB2. So adapter protein. That's GRAB2. In case you forgot. Binds RTK, STAT, binds receptor, then it's self-to-form dimer, PA3 kinase. These are all examples of what have SH2 domains. So if anything's messed with that phosphorylated tyrosine, which we'll talk about in a second, it's not going to bind, right? SH3 is the domain that binds a PXX motif on the other protein, okay? So remember, SH2 domains bind to proteins that have the phosphotyrosine binding. It doesn't have it itself. Same with SH3. It's going to bind a PXX motif on something else. It doesn't have it, okay? So when things grab onto each other, like GRAB2 binds SOS, SOS has the PXX motif, to which the SH3 domain in GRAB2 binds to. So it's grabbing onto something else, and it's the same for the opposite way around, okay? So remember, the domains, they don't actually contain the phospho-binding domains. They don't contain the phosphorylated tyrosines. They grab onto something that has that. Same with the SH3. It's going to grab onto a protein that has the PXS motif. Yeah? Okay, JAK-STAT. We're also kind of going to go through this like relatively quickly. So prolactin, you don't have to know that it's prolactin. Signal binds the receptor, causing the dimerization of JAK1/2. What does dimerization means? 2 becomes 1, right? Causing the cross-phosphorylation of each other, also known as what? What's the other name for it? Autophosphorylation. So now they're active, and they activate the intracellular portion of the receptor. What does intracellular mean? We're in the cell, okay? It's going to activate the intracellular portion of the receptor, tyrosine kinase. So this is really important. STAT proteins have the SH2 domain, right? That binds to the phosphorylated tyrosine kinase, right? So again, it's going to have the SH2 domain. It's going to bind to the phosphotyrosine kinase. Okay, it's important to have that distinction. Which is going to cause the phosphorylation of STAT. STAT is these guys. Okay, so cross phosphorylation that activates STAT, active STAT. That's what that is. The STAT proteins are going to dimerize through the SH2 domains. So again, 2 becomes 1, dimerization. And then it is important to nucleus to act as a transcription factor for milk protein production. Who remembers what a transcription factor does? What does it do? How? How does it activate transcription? Close. A little bit more specific. Who remembers what a transcription factor does? Yeah, so the RNA polymerase, I think, right? RNA polymerase is not important, but it basically tells it, transcription factor tells it exactly where to go to start transcription. Otherwise, it's not going to know where to bind. So, okay, I don't know why the question's already on here, but if there was a loss of function mutation in the phosphorylatable region of the JAKs, so JAK is just another kinase, just another kinase 1, just another kinase 2. If there's a loss of function mutation in that region so that these regions can't be phosphorylated, the pathway is going to stop because, let's go back to here, right? So signal binds, they come together and they cross-phosphorylate each other. If they can't cross-phosphorylate each other, is that signal going to get through? No, right? So loss of function in the phosphorylatable regions of the JAK so that those regions cannot be phosphorylated. It just means that if these guys can't be phosphorylated, then what are we going to put a phosphate onto, meaning we can't do anything? Yeah.
Related to like things like where like affects this proportion, like I know you're gonna, we should talk about this, but do we also have to like then go on and explain like further about like the steps afterwards, so it's like for like more detailed explanation, or it's just like having an explanation?
On her exams, explanation is always better over deletion. Like always, just if you don't know what it is, like there used to be so questions that I would like look at and I didn't even know where to start, and I would just start writing as much as you can because you can always get partial credit, which is what I like about her exams. But I wouldn't go too crazy with like the overall process. Don't overthink it, guys. On this exam, there's gonna be so much information in your head. Don't overthink it. Don't talk about everything except for answering the question. We'd rather have you answer the actual question because we're the UTAs who grade it. Answer the actual question and then give me the explanation. Don't dance around it. Just try to answer it directly the best you can and then give me all the explanation. But don't get too much in the specifics. But when it comes to things like survival signaling, so like bad letting go of BCL2, I want you to write that over and over again. So we will have the cell live because bad let go of BCL2. Keep saying that. I don't care if it's the same question, they ask you the same thing over and over. Keep writing that, okay? Always have like that little, okay, the cell is going to survive, but why? Because the ending is what's the most important, right? If bad stays to BCL2, it's going to die. Like that's what determines everything. So make sure that even if she asks you over and over again, you have those kinds of important details necessary. Does that make sense? Did you have a question?
Wondering why the like signaling molecule where this one isn't a dimer, it's like for all the others, it's like the shape of it that brings the receptor together, right? But this one isn't.
Are you saying the signal is a dimer or is not a dimer?
Doesn't
Yeah.
get it.
Yeah. So there's so many different signals for so many different things that they can come in like all shapes and sizes. So you're gonna have pathways that are gonna have a certain type of signal. You're gonna have this one that isn't. And that kind of looks weird. It's prolactin, so it's a little different. But don't worry too much about the specifics with the signals themselves. We more care about what happens in the pathway. Okay, this is important. So if you mutate the tyrosine in the receptor, the one that is phosphorylated by just another kinase, right? So there's a part of here that gets phosphorylated by JAK. Like they come together, they phosphorylate each other. So if we change that tyrosine to alanine, which can't be phosphorylated. She will ask you something about this. What can no longer happen? What's going to happen? Yeah. Yes. And why? What do SH2 domains need to bind to? Phosphorylated tyrosines. Okay. That's really important. She may ask you a question. I know she did this one year. She was like, you have an experiment and you want to figure out which of the two like jacks are working, like two of those regions are working and one of them's not working. You want to figure out which one's not working. How would you figure that out? What I would say is change one of them at a time to an alanine and see what happens, right? So if alanine can't be phosphorylated, then STATs can't bind to the receptor because the SH2 domain needs to bind onto the phosphorylated tyrosines. Okay? We okay? Anyone have any questions about that? Anyone still wants me to re-explain it? Yeah.
Explain what the question that you mentioned was. I was just kind of confused by that.
Oh yeah. It's I might have had it in an old presentation, but it was like, oh, something's wrong. We know that maybe something's wrong with the two guys at the beginning, right? The two Jack subunits. How would we figure out which one of them is wrong? Or like what's going on there? A question you could say, an answer you could say is like, well, if we change the tyrosines that need to be phosphorylated, change one of them to an alanine, and it's still not working. I mean, it's not working, but also when it has the tyrosine, it's still not working, then that's something's wrong there. Don't think too much about that. Okay, so same question. So we say that it's changed to an alanine. Will the signal still cause the receptor to dimerase? What do you guys think? Anyone? So signal comes in, they dimerize, cross-phosphorylate. The two regions can't phosphorylate each other anymore. Can the signal still cause the dimerization? Yes or no? Yes. Yes. Yes, it does. So that part, the dimerization is dependent on the receptor binding the signal. Receptor binds signal, two things come together, then they cross-phosphorylate each other. So if we change that third step, if we change that tyrosine to an alanine, not making it be able to be phosphorylated, the signal will still bind to the receptor. That doesn't matter. That make sense? Okay, let's do a little recall. So, what are GEFs? Can somebody tell me? Let's get this out of the way. What does it stand for? Does anyone know what it stands for? Yeah, I had to Google it today. So, guanine nucleotide exchange factors. So they work with the G proteins. We'll talk about the GCPR in a second. So, what I want you to remember is, okay, I can't write anything, but GEFs exchange GDP for GTP. What do we say about GTP? When we see GTP, is it active or inactive usually in general? Active, right? I made the mistake on the slides. I'm going to correct it, but active, right? So that's how I remember them, okay? That's really important to know because if you get them mixed up, then you might get a question wrong, which I think there was like a question where like people got it mixed up. So I really want to go over this again because it's really easy to get these two confused. GEFs, GTP, GTP, turns it on. GEFs turn on, okay? GAPs, GTPase activating proteins. I don't really have a memory trick for this because GTPase confused me in my head and it takes off a phosphate, so that kind of confuses me. But GAPs take off the phosphate, so exchange GTP for GTP, which turns it off, okay? GEFs turn it on, GAPs turn off. Good? So, the induces GTP hydrolysis. That's like the fancy thing. Hydrolysis is just when we take a phosphate off. That's just all it means. Okay? Yeah.
Is there a word that's like hydrolysis but for yes?
I think that is the term. No, because they actually they like exchange it for the GTP. So since they're adding a phosphate, like a term like hydrolysis.
Yeah.
So it's weird because it can like make stuff lose affinity so that GTP just diffuses in, not necessarily like a kinase, which like adds stuff. So just remember that GEFs exchange the GDP to GTP. Okay.
So like when you have like GDP, it's not adding a phosphate to make it GTP. Your GDP is literally just diffusing out so your GTP can diffuse in. So you're not adding a phosphate group onto that GDP. Does that make sense?
Unless there's like a kinase. Kinases
Yeah.
like add phosphates, so that's why you would see the change there from GDP to GTP. But in this case, with GEFs,
With
and GAP,
gaps, yeah.
it just literally comes out and then the other one comes in. And so usually these two guys help the chemistry change so that something loses affinity for GDP or something loses affinity for GTP to let go of it. Does that make sense? So GCPR, this is confusing to people. It's a lot simpler than you think. So whole thing that happens, the GCPR, I wrote that wrong, is just the squiggly. It's just that. That's all it is. So signal binds, conformational change happens. And when the structure changes, what else changes? Function, right? Changes the intracellular receptor. Intracellular means inside or outside the cell. Inside. It changes that conformation, and basically it just results in GA, so blue guy, releasing from his brothers B and Y, beta and I forget what that last one is. So they separate, it's diffusing away. And now, because that chemistry has changed in GA that made it detached from those two, GTP can now diffuse in. So it releases the GDP. Remember, the whole thing is just kind of floating away somewhere. GTP can come and diffuse in because now GA has changed its chemistry. So now that it likes GTP. And now, is it on or off? It's on, right? GTP on, GDP off. Okay, so it's active. That's the bottom here, right? Bottom, active. And now it can like do its thing, signaling thing. And then GA is going to bind the effector protein. Effector protein is just these green guys. Don't have to know the specifics about that. Just know that when it diffuses away, that's just like what's holding on to it, basically. And GA hydrolyzes GTP. What is hydrolysis? Are we adding or removing a phosphate? Removing. Hydrolysis, removing a phosphate. So it hydrolyzes GDP. So GDP is coming out. Coming out. We're going to talk about RGS in a second because that was something on your quiz that like a lot of people didn't know. We'll get into that. But basically, GTP comes out, and what gets to come back in? GDP, right? So it does its thing. It's like, I think that's like a phosphate. Let's go with that with the help of RGS, which we'll talk in a second. GDP can now come in, and now it's resting. Now it's done. Because we don't want it to be active all the time. We don't want it to be active all the time, right? It wants to do its thing. We want it to be done. Yeah.
You need to know like what it does to the effector protein. Like, does it add a phosphate?
No, it just binds it. It's just holding on to it. So like when these two guys separate, it's like this is gonna hold on to it.
Okay.
And then we'll talk about like lipid anchors, which she mentioned once. So question on your quiz, see how she mentioned this twice. This was from the problem set. This was from the quiz. She's really obsessed with this. I don't know why. She is. We're going to talk about it. We're going to go through it. We're going to get it down. Okay. You guys are going to leave this review session and you're going to be so happy. You're not going to be happy, but you're going to, I hope you feel a little bit better after this. So let's talk about some similarities. So this was, again, one of your quiz problem set questions, really confusing. I had to Google stuff. What are some similarities between RAS-RAN behavior and GA? Remember, GA, blue guy. Blue guy. And RAS-RAN is... RAS-RAN is... RAS-RAN. It's over here. RAS. Do we see any similarities? Do we see any similarities after looking at the two diagrams? What do we think? Anyone? Yeah. Yeah, right? So it needs GTP to be on, and it needs GDP to be what? right? That's a similarity, right? That's what's making the active/inactive happen. That's really important. What else?
We'll have gaps and gaps.
Interesting point. So, okay. So this was a, we're gonna get into this in the next slide, but I'm glad that you said that because I'm gonna address that. So RAS is activated, big red circle, so you can see, on or off based on the presence of either GDP or GTP, right? We talked about that. So GTP on, GDP off, right? When GA binds to GDP, it's in an active bound state. When the hydrolysis happens, which means we're taking off around a phosphate, off, right? Hydrolysis taking off. I'm sorry, not taking off phosphate, but it's getting out. We're getting rid of GTP. It's inactive because GDP diffuses in. They both need GEF activity. Remember, GEF is when GDP diffuses out and GTP comes in. But the opposite way around doesn't go. So that's the difference, okay? GA doesn't actually have a GA GEF, right? It's just the GCPR. So when the signal binds, the conformation changes that GA can separate from the two, let go of GDP to let GTP in. That's its RAS GEF. This was the answer to those quick questions. This is really important to know. The GEF in this case is just the GCPR because that's what's changing the conformation of everything, right? It's not like a molecule on its own. So the second thing is RAS-RAN use a gap to help with GTPase activity. So that just means RAS-RAN uses a gap to help it turn it off. We do not have that here. Instead, we have the RGS protein. So that light blue thing over there. It basically helps us speed up hydrolysis. It lets us speed up the process of kicking GTP out so GDP can come in. So, and a couple things she also included, sometimes they can have their own intrinsic rate of hydrolysis and they don't even need one. So it's just like a little helper guy sometimes. And then sometimes a downstream effector protein can play that role too. So sometimes the down one can like come out and help out with hydrolysis. So if you're confused on what RGS is, just a little helper protein. Sometimes we need it, sometimes we don't. It just helps us speed up the rate of GTP hydrolysis, which means we're speeding up the rate of how fast we can let go of GTP to get back GDP, which means is the pathway back to being on or off. GDP is there, which means it's off. I know I went through that fast, but does that make sense? Does anyone want me to go over it again? Okay. Okay, important takeaways. I wrote that wrong again. Okay. It acts as the GEF for GA, meaning that's what's letting us go from off to on. Okay. GA is a GTPase. GA is a GTPase. That is why it doesn't really need much help. It can use RGS, but it can pretty much just do it on its own if it needs to, which means the GTPase is just something, it's an enzyme that helps speed up the hydrolysis of GTP. Okay, so that's really important. So the similarity there is they both need GEF activity. They both need to be turned on. They both need that. The difference is when they're turning off, one of them has a gap, which is the RAS-RAN, and one of them has the RGS, or it can just do it itself. So that's that question kind of broken down. Does that make sense to everybody, the difference between RAS-RAN activity and GA and RGS? Do we know what RGS is now? Okay. I just like ignored the last two. If the signal is not bound to ligand, GB can bind and activates them. If it's still bound, it can reactivate. I don't really think she's gonna like go into too detail about that, but I do think she's been real sus with like how much she's posting this question. So I feel like we need to go over the GEF and GAP again because those are easy to mix up. It's easier than you think. These are just fancy terms for simple things. Okay. Okay. Microtubules. We found through topic 5. Okay, before we move on, does anyone have any like lingering questions? Yeah. About anything we just talked about? We can go over stuff again. Yeah.
She's probably not gonna give us the pathways, like diagrams, right? We didn't get it from
No.
those.
No, I don't think she's going to.
Ooh. Would she give them like a diagram without the labels? Like she does
I don't think so.
sucks, guys.
Yeah, I would just really know the pathways because I don't think you're gonna have diagrams, so.
Just if you just got to memorize the steps, like step by step, so that because all of our questions about pathways is like, if this one random thing is changed, what's going to happen? If you have the steps down, which most of them are just like, oh, this activates something, which activates something. Those are like majority of the steps. You'll be okay.
Oh, I'm gonna go in yours. Okay, cytoskeleton overview. So we have actin, microtubules, and then intermediate filament. So we really only go over microtubules, so that's what this is going to be focusing on more. So basically, cells have all three. It helps with the structure. Okay, so actin. Basically, actin, use it for muscle contraction, cell mobility. They serve as a track for a motor protein called myosin, which is also used when you like move your muscles. Yeah, that's actin. And then their polymers are made up of actin subunits. So that's what it looks like. Okay. The cytoskeleton also has intermediate filaments. So basically those are mainly used for their structure. And then they maintain the shape of the cell and the shape of organelle. So basically you don't want your cell like shrinking and like changing its shape. You want it to have like a good shape. So that's where your intermediate filaments come in. And then we have our microtubules. So our microtubules resist compression, and then they provide a track which vesicles move. So like your kinesins and your dyneins, like they walk on your microtubule track. Microtubules are also important because they're used in cell division. They are the things that pull your chromosomes to opposite sides of the cell. So they can eventually, like one cell can become two cells. They're structural elements of flagella, cilia, and centrioles. So like flagella and cilia basically just like help a cell move. And then they are specific to eukaryotes. So only eukaryotes have these, and then prokaryotes have something different. Does anyone have any questions so far? No? What's up?
I'm not sure what to know about actin or intermediate filaments.
I would know that they're in the cytoskeleton, but I would more focus on microtubules. I think she's gonna specifically be asking about microtubules. Okay, microtubule structure. So microtubules are your polymers, and then your monomers are gonna be tubulin. And then tubulin is made up of your alpha subunit and a beta subunit. Both subunits can bind GTP, but only the beta subunit can hydrolyze your GTP to GDP. This makes tubulin a GTPase, and then microtubules are hollow. So if you like look at this picture right here, they're basically like making this, but the inside is hollow. Does that make sense? Okay. Microtubules are polar structures where one end does not look like the other. So basically your microtubules have a plus end and a minus end, and there's going to be different activity happening at your plus and minus ends. So your minus end has the exposed alpha subunit of the tubulin, and then your plus end is the exposed beta subunit. So your minus end is what's going to be in like your MTOC, your microtubule organizing center. And then the plus end is what we focus on when we talk about like polymerization, depolymerization. And then so tubulin addition and subtraction can happen at either end, but it's much faster at the plus end. Okay, so basically if you look at here, you have your tubulin dimers, dimers 2, you have two things, and then this is just showing like how your microtubule is formed. So you see it goes like 2, and then you have they all like combine together to make your microtubule. Okay. So there's something pretty cool called fluorescence microscopy. So basically we can see like cytoskeletons, we can see microtubules. you basically tag tubulin with GFP. Basically, what GFP is, it's green fluorescent protein. So when you use fluorescence microscopy, sorry, you can see like your microtubules. So if you look at the green places right here, they're tagged with GFP, green fluorescent protein. So yeah, anything in green is gonna be your microtubule. Anything in blue is gonna be your DNA, and then anything in red is gonna be active. So basically, yeah, you're able to see it. And then, okay, one function of microtubules is that they're used to transport organelles. So basically, with like your kinesins and dyneins, they separate chromosomes during M phase, and then they're important for cilia and flagella. And then just a little note right here, tubulin can only be added to a microtubule if it's in the GTP bound state. So if it's in a GDP bound state, it's not going to be added to your microtubule. Okay. Dynamic instability. So basically your microtubules are constantly growing and shrinking, and it's based on the addition of tubulin to the beta subunit and GTP hydrolysis at the plus end. So your minus end is going to be in your microtubule organizing center. And then, so basically, in order for your microtubule to grow, you have to have tubulin GTP added onto the plus end to create your GTP cap. So basically that like happens during polymerization. And then, yeah, so for it to grow, you have to have your GTP cap. And then, okay, so if you look here, if this is your plus end, so your addition is going to happen much faster at the plus end. So basically your microtubule, it's always going to be growing and shrinking. So right here in this middle, you're going to have GTP hydrolysis. So that means your GTP is going to lose a phosphate and it's going to become a GDP. So that's happening in the middle. Over here at your plus end, you're going to have your cap, and then you're going to have more tubulin GTP added. So basically what a catastrophe is if your rate of GDP hydrolysis in the middle, because if you have GDP bound, it's going to start to fringe off. So basically like a catastrophe is when you have more tubulin in your GDP bound state, and then it's going to rapidly shrink. Does that make sense? Okay, and then your rescue is you're going to have your microtubule grow after it shrinks. Okay. This was on your quiz, so I'm going to have you like read the question, talk about it with your table for like 2 minutes, and then we'll go over it. You want me to do this? Somehow it makes sense in my head, and then when I explain it out loud.
No, like, but that's kind of focus. Like, it's not as easy as like, I feel like Soul Cycling is like, this does this, but like, to those like, you want me to say the answer to the question? Or do you want to do the answer? Let's see. It's like the one that was like, if you sever it in the middle, it's gonna. You feel comfortable doing it? Or you want it?
Yeah. Yeah, it makes sense in my head, and then when I get it out, it's like no. Okay, does everyone have like an idea of what's happening in their head? Who can, okay. So
What do you guys think? So if we sever the microtubule in the middle,
What do you think is gonna
what's
happen?
gonna happen?
Is it gonna grow or is it gonna shrink? Someone tell me.
I thought it was gonna shrink because in the middle of the microtubule,
Mhm.
all that tubulin is being hydrolyzed to GDP.
Mhm.
So if you cut it in the middle, that tubulin bound to GDP will shrink because that's what it does.
Exactly. Okay, so if you sever a microtubule in the middle, you're gonna generate new plus and minus ends. If you have new plus and minus ends, do you think there's gonna be a GTP cap? Yes or no? No, right? If you do not have a GTP cap, do you think your microtubule is gonna grow or shrink? Someone just raise your hand, tell me. Right, it's gonna shrink. So you don't have a GTP cap on your new end. So that means that your tubulin in the middle of the microtubule is already gonna have hydrolyzed its GTP to GDP. And then when you have your GDP, that's when it's gonna shrink.
Okay, let's go back to that photo that you put into. Yeah, so shrinking, right? So this is tubulin to GTP, right? And things are keep being added. Once that happens, GTP hydrolysis, which is just like the green and the dark green, if that doesn't have protection, it's not the GTP cap, it's just gonna shrink. So if you cut in half, new ends, those ends don't have the GTP cap that we want because everything's hydrolysizing. I mean, hydrolysis happening as you keep going along and adding new stuff. So it's gonna shrink. Yeah.
She said also that it could be like different rates of hydrolysis. So I think it could be, so based on that question, I was reading it as if, there would be the GTP cap where they cut it. Because
Yeah, no, I get it. So that's a good point. The thing is, what I would say is, so there's new ends for both plus and minus. So since what do we add? Tubulin being added, which end does it go onto? Plus end, right? So that's when it's being added, which is where it would bind to GTP. So if you cut it in the middle and there are those new ends, but tubulin is still adding away, it doesn't have that GTP cap, so it's gonna hydrolysis. Like hydrolysis is gonna happen. Is that
it's not a GTP. Like at the
it's because it's as it's adding to the microtubule, the tubulin, the GTP bound to the tubulin is gonna hydrolyze over time from that middle. So as stuff is being added, hydrolysis is happening at the same time. So it's trying to keep up. So tubulin's being added, hydrolysis is happening. If the hydrolysis rate overtakes how much tubulin's being added, then things are going to shrink, right? So that's, I think, what she means when like the rates. If you don't do enough tubulin, if you don't do it in time, the hydrolysis is going to catch up to it and it's going to shrink. So if we have one, tubulin's being added, cut it in the middle, new ends are generated. That doesn't have that GTP cap that we had before. That's just on its own now because it's not the new thing that's being added to. So it would shrink. Does that make more sense?
Okay. MTOC, who can raise your hand and tell me what it is? What does it mean? Exactly. So your MTOC is your microtubule organizing center. So basically, if you're in a cell, it is super hard to create a microtubule from nothing. It needs a base to start building off of. So that's what your MTOC is going to be. So your MTOC is your base for the microtubule to build off of. And then microtubule nucleation occurs in 2 steps from the centrosome. So you have a gamma tubulin ring complex. So then you have a gamma tubulin small complex assembly. So basically, what that is saying is your minus end is going to go into your microtubule organizing center, and then that's going to be your base to add your tubulin to the plus end. So just like remember, like it's super hard for microtubules to just build off of nothing and start adding tubulin together. So your MTOC is basically a base to make that easier to happen. Does that make sense? Any questions about that? What's up?
So there's like the centromere, and then there's the basal body. What's the centriole? Because I know you had mentioned like centriole before. Isn't centriole like inside of the centriole?
I don't know. That is a good question.
Yeah, I don't really know. Not such a-
If you don't need to know.
I think it's for prokaryotes. It's the MTOC for prokaryotes.
Mm. Yes. Yeah, we just hit Google real quick.
Centriole makes spindle fibers, according to Google.
Animal cells. We'll come back to that. We'll figure that out when we come back to it.
Okay, so microtubules in vitro. So basically, this is your graph. If you have tubulin, you have a test tube, and then you measure how much those microtubules come or how like it forms. So basically, you have 3 phases. You have your lag phase, your elongation phase, and your plateau phase. So if I go right here, what phase is this? Lag phase, good. If I go this, what is this phase? Good. And then what's this? Good. So you have 3 phases. Let's see.
Oh, yeah, real quick.
Okay.
So, there was a quiz question that was about lag phase. So it's like, how do you abolish lag phase? The reason we have lag phase is because it takes time for them to form from nothing. They need like a base. So does anyone have an answer to that question of how would you abolish lag phase? So not speed it up. How would you abolish lag phase? Yeah.
Oh, not to speed it up.
Well, like speed up, it's basically nothing. Like you don't.
Get amount of it, but then you could also add like drugs that could completely get rid of it.
Right. So that's important. So Taxol would be an answer to that question, right? Which we'll talk about. But MTOCs, what she was saying before, like they need a base to start building off of. So if they don't have that already, they're going to take time to assemble and then to bind to that to start making the microtubule. So if you have the MTOC there already, if you have Taxol, a lot of Taxol, if you have, what is it, the gamma ring, whatever complex thing, like just having a base is really good for abolishing the lag phase. If any of you were confused on that question. Does that make sense?
Okay. Your 3 phases. So your lag phase. Your lag phase is going to be that very first part of the graph. And what that is showing is that it takes time for a microtubule to form from nothing. So basically in that lag phase, you have your tubulin, you have like your microtubule organizing center, and that's like the start of it. So your lag phase is like when it's just starting to form. And then your elongation phase, once the seeds are formed, your microtubule elongates at a much quicker rate. So your tubulin is being added to the plus end rapidly. So that's why if you see like here, it's kind of steep. That's why it's going up so quickly because your microtubule is growing a lot. You have a lot of tubulin, GTP, and then that's all being added to the plus end. And then your plateau phase. So during your plateau phase, your rates of polymerization and depolymerization are going to be the same. They're going to be equal. So the rate that you're adding tubulin GTP to your plus end is the same rate that you have your hydrolysis in the middle. Does anyone have any questions about that? Okay. And then this is in vitro. So what this means that if left long enough, the tubulin will complete, or it's going to be completely, your GTP is going to be completely hydrolyzed. Is that a word? Hydrolyzed to GDP since it's a closed system. So eventually, like your rate of hydrolysis is just going to hydrolyze all your GTP to GDP. Okay. microtubule, and drugs. Okay, so you have Taxol. So what Taxol does is it increases tubulin's affinity for other tubulin, which stabilizes your microtubule. And then you can also prevent depolymerization. If your microtubules are super stable, they're not going to be able to like depolymerize. Taxol is also used as a chemotherapy drug, so it prevents your spindle assembly. So basically what cancer needs is dividing cells. So cancer cells need dynamic instability in order to like separate. But if you stabilize those cells, then they're not going to be able to divide. So that's why Taxol's commonly used as a chemotherapy drug. Okay, and then Colisomid, is that how you say it? Basically what it does. I would know Taxol the most. I would review these, but I would definitely just know Taxol. What's up?
It wouldn't abolish. So the main answer she was looking for in that question was the MTOC and the like, you can even add like microtubule seeds. But Taxol stops things from shrinking. So by adding it, it would basically like help speed up like building that base. Like that is part of it, but that main answer she was looking for to like do like the most that we possibly can is MTOC. Yeah.
So like, that was on the exam for Taxol still, right?
Yeah, so on her answer key for that quiz, she had only like MTOC and seeds, but then she was like, if they have Taxol, like I'm okay with that answer as well. But to be safe, I would mention MTOC because it's just like it just needs a base. Like that whole lag phase is about building the base and getting ready, and MTOCs help with that.
What's up?
So like the gamma tubulin ring complex, would that count too?
Yeah. Just base. Something that we can start with.
Okay, so this is a graph. I took this directly from like the answer key on the problem set. So Taxol is a microtubule stabilizer. So you see polymerization, but you don't see depolymerization. So basically right here, this is going to be your graph when you have Taxol. So if you look at it, your lag phase is really, really small here. You have a small lag phase. You have a quick elongation phase because when your microtubules are stabilized, it's a lot easier to add tubulin GTP to that plus N, so they can increase. And then you are gonna have a plateau phase with Taxol. So don't think they're just gonna like, if you have Taxol, they're just gonna grow infinitely. You are gonna have a plateau phase.
Oh yeah, and I will say like on when I took this class on the exam, I think she definitely had some questions about like the graph specifically.
Yeah.
Like if you add Taxol, like draw the graph what that looked like, and that would just be like the same, like slightly shorter lag phase, steeper elongation, and still having that plateau phase. So it's good that she said that Jen just said like there is still a plateau phase because some people just assume okay like we're good to go, you know, things are never going to shrink, we're chilling, but it's still eventually going to reach that plateau phase. Yeah.
Why would there be bigger, like, why would it go up on the graph? Isn't there the same amount of like tubulin GTP? So shouldn't it be around the same?
So what's being added, right? So there's GTP hydrolysis that's happening at the same time as things are being added. So let's say like that rate of stuff being added on was pretty slow. Like they're still being added on, but it's pretty slow. Taxol is going to come in and say, hey, we're going to pause the GTP hydrolysis for now. Like we're going to slow it down so that you can have time to keep adding stuff on. So that's why on the graph, it would be steeper. It's just, it's the total amount of tubulin, right? So it's like plus the GTP hydrolysis and the stuff that's being added. If we put a pause on that, we're going to have more in the microtubule in general, right? Because that stuff isn't coming off as much anymore. Taxol is kind of stopping that. So we're still adding stuff. Does that make sense a little bit more?
Yeah.
It's like the total.
Because in the when it's GDP only, there's some hydrolysis that removes.
Yeah, so like GTP hydrolysis is happening as stuff is being added on. That's always still happening. Taxol comes in and says, hey, we're going to stabilize, which means we don't want any depolymerization, which means we don't want things to shrink. So it stops it. It's kind of like a pause button. It's like, we're going to pause that for a second. We're going to still keep adding, but now there's less stuff like being thrown off. And so overall, there's a bigger microtubule because there's less stuff being thrown. Is that better? Kind of.
Okay, microtubule associated proteins. So you have a protein called tau. It binds to microtubules and it stabilizes them. Kind of the same thing as Taxol. Like it binds to microtubules, stabilizes them. You don't have depolymerization. These are abundant in the central nervous system, and kinases can phosphorylate tau, and it will dissociate away from your microtubule. So like your kinase comes in, phosphorylates it, and then tau dissociates. Once it dissociates, it can become like entangled in the cell, and that's where it starts to cause issues. And dementia, Alzheimer's are thought to be caused by like dysfunctional tau because it's obviously in places in the cell where it shouldn't be. Does anyone have any questions on that? What's up?
Talk about the previous slide, the, if you did a graph of like there was a question on the problem set for like this graph and then also a graph of if you added a bunch of would it look pretty much the same with the high
Yes.
concentration?
This was the graph from that question. I just wanted to show you guys that like Taxol, like it lowered the lab phase. But yeah, this is also like if you had a bunch of like GTP.
But also, like, sometimes she'll throw in, I think this is also on your quiz, like, there's GTP tubulin that you could add. And then she's like, what's the solution? You could add non-hydrolyzable GTP tubulin. That is like a thing that she mentioned. So you can still have more GTP tubulin, which would help still. But if you have GTP tubulin also that doesn't hydrolyze, that's also really helpful. And that's like an actual thing that you can do. So.
Okay. If you perform an experiment, but you use ATP instead of GTP, what will the effect be? Which phases won't occur? Will any phases occur? What do you guys think? Take like a minute and talk to your table about it.
I don't know. Oh my god, fire question.
I took it from one of the other.
No, I love it. It's like a Voltron question. I was thinking of the one from the quiz. Was that Kinesin walking or something? She was like, I forgot ATP. You know that
Yes.
question?
Yeah.
What was it?
It was kind
Oh,
of
okay. Like a walking thing. Or whatever.
Yeah.
It's a specific question. I'm learning right now. You're killing it. You're killing it.
Okay. Who can tell me what's gonna happen if you have ATP instead of GTP?
So instead of GTP binding to tubulin, it's ATP now. What's gonna happen?
What's gonna happen? Someone tell me.
If there's no GTP, then there's no stability available for the dimers to bind together, so one of the things is
You are 100% correct. Good. So no phase will occur. This is because tubulin can only be added to a microtubule if it's bound to GTP. So if it's bound to ATP, nothing is going to happen. No phases will occur. Your microtubule is not going to polymerize at all. Does that make sense? Okay. Okay. Microtubule associated proteins. So you have kinesins and dyneins. Kinesins walk towards the plus end, and then you have a couple of different ones. So the first one we're going to talk about is kinesin 13. So if you look right here, they have their motor domain is in the middle of the protein, and they actually do not have directed movement, but they slide. So if you look at this, your kinesin 13 is going to slide to the plus end of the microtubule, and it uses its ATPase activity or domain to rip tubulin off the plus end, and it actually destabilizes plus end. Does anyone have any questions about that one? So that one's not walking, that one's like, it's sliding. And then it's gonna rip tubulin off the plus end and it's gonna destabilize it. Yeah.
We'll get into that. We have some questions about that too. I would say more like group them into like what they do. Like I guess not the super specifics, but they each have like a very specific function. And a lot of those like kinase families can be grouped together. So yeah.
No like basic stuff about each of them, I would say. Okay, and then you have your kinesin 3, so that's involved in cargo transport in neurons, specifically neurons. So that's different from other kinesins that are just transporting in the cell. This is for like neurons.
And dimer, right? That's like there's two guys here. So there's two pairs. That's what the dimer means. Works as a dimer.
Okay. And then kinesin 14. So you learn that all kinesins go towards the plus end. This is going to be your one exception. So kinesin 14s walk towards the minus end of microtubules. And they're, yeah, that's it. Basically what they do. They walk towards the minus end. They're the one exception. Okay, mechanism of kinesin walking. You guys can read this, but basically kinesins diffuse around. When a motorhead makes contact with the microtubule, it changes conformation. ADP dissociates, allowing for rapid ATP association. The motorhead has good affinity for the microtubule. ATP binding causes the neck linker region to swing the other motor domain forward. The one that just swung forward contacts the microtubule, which causes ADP to release an ATP binding. And then in the trailing motorhead, ATP is hydrolyzed to ADP and the motorhead releases from the microtubule. ATP binding in that forward motorhead causes the neck linker region to swing the other trailing motorhead forward. I'm not sure how much you need to know this in detail.
So like there was a question on your, I think it was the quiz, right? I think it was, oh yeah, it was the quiz. It was like,
Yeah.
oh, if it can't walk anymore, what did I forget to add? And it was like, I think it was something like that.
Yeah.
Yeah, I think just know that it keeps switching back and forth from ATP to ADP as it's walking.
Mhm.
And as usual, like structure determines function. So you change the chemistry, it's gonna do something else, gonna move and link onto other things. But I would say like don't get too wrapped up
Yeah.
in this.
Just know that it needs ATP to walk. If it doesn't have ATP, it can't walk.
And it also needs to change too between both of them to like keep doing the movement.
Okay. Back to kinesins. You have kinesin 5, which is a tetramer. So basically you have 2 heads and then 2 tails on each end. One example is egg 5, which I know that she mentioned specifically. This can cause overlap of 2 different microtubules. So if you look at this is one microtubule and this is the other. So it causes overlap. That's important in spindle assembly. And then it stabilizes the microtubules and controls like their movement in respect to one another. And then you have kinesin 1. So basically this transports cargo in the cell. So if you see this, these are your heads and then your tail is going to be attached to your cargo protein. Works in a dimer formation. And then you have 2 heads and then 2 tails and it's plus end directed.
Okay.
Okay. Which kinesin families are dimers that walk along the microtubules carrying cargo? Talk amongst yourself for like 1 minute and then we'll come back. I'll take these from the other review slides.
I added this from the quiz. I think quiz, no, it's problem solving. Yeah, that's why there's so many slides because I kept like doing it.
I feel it.
I think we'll be done, like, I think we're kind of gonna speed through the cell cycle stuff. Let's talk about the
Yeah.
restriction period. Just like, we can speed up. We can just be like, you guys can do this. Whatever. I can take over at cell cycle.
Okay, do you want
To just like speed through it.
Okay. Okay, who can tell me which families of kinesins?
This was a problem set question.
How it was 1 and 3?
Good. It is 1 and 3. So kinesin 1 transports cargo around the cell, and then kinesin 3 is going to be the one that transports cargo around the neuron.
So your question over here earlier about like what you should know, like these guys both transport cargo. So like you don't, but like it's not asking about like specifics of structure or anything. So like know the job kind of each. And we just like add this in. I don't know if she's gonna get into specifics of like where it transports cargo, but 1 is in the cell just generally, and then 3 is in a neuron. She said this, guys.
Okay, which kinesin family doesn't walk along the microtubule at all, and why? Someone can just like raise their hand and tell me.
Shout out the number. Shout out the number.
13. Good. So, kinesin 13s, these are the ones that don't walk. They like diffuse to the plus end, and then they destabilize the plus end.
I don't know why it says used.
Oh, used during anaphase to shorten kinetochore microtubules. You guys don't need to know that, but just know that they diffuse along to the plus end, and then they destabilize the plus end.
Okay, we're kind of gonna go a little bit faster through cell division, cell cycle stuff, part 1. We'll just mostly talk about like restriction point stuff, and the mechanism of that. So again, we're not gonna like teach you guys from scratch, but we're kind of gonna go through this. So G1, cells receive the growth factor signal, starts thinking about cell division, and the committing to the cell cycle happens in late G1. So which point happens in late G1? Restriction point. So why do we need a commitment point? Why do we need a restriction point? Because if there's something wrong, if there's DNA damage going on, we don't want the cell to keep going. We don't want the cell to keep making itself, right? So restriction point happens at that late G1. That's the commitment. S phase, DNA replication. If they can't go through, they will go through the cycle, they'll die, and they can halt replication. G2, the cells keep growing because G stands for what? Grow. They're going to grow again once ready to go to M phase, and a cascade of events happen. And this is the second checkpoint, which is G2. So if it's not ready to keep going, it will inhibit entry into M phase until that cell has grown enough. So we have 2 points now. What was the first one? Restriction. So we check for damage, check if everything's okay to keep going. Now we have the second one, which is G2. It hasn't grown enough. M phase, this is where chromosome segregation happens, spindle assembly. And this is where cytokinesis finishes the cell cycle. So she loves this checkpoint. So SAC, spindle assembly checkpoint. So it inhibits anaphase, it inhibits the segregation, right? The two coming apart until all the duplicated chromosomes have the proper microtubule attachments in the spindle. She really likes that checkpoint. She also really likes the restriction, which we'll get into. So if you added a drug to dividing cells that were able to stabilize the microtubules, what phase of the cell cycle would your cells arrest in? Anyone have any guesses? Anyone have any guesses? Yeah.
M phase, because it would mess with the like depolymerization of the microtubules, which is needed in creation of strand.
Right. Microtubules are associated with what assembly? Spindle assembly. Microtubules are associated with spindle assembly. If there's no dynamic instability, which means what? Can somebody remind me what dynamic instability is? There's growing and shrinking. That's what that means. Cell division will stop and the cell will undergo apoptosis. Apoptosis is what? Cell death. And if it arrests for too long. Does that make sense? So microtubules associate that spindle assembly. And that G2 checkpoint. So G2/M, I think M, I think microtubule. Okay, why are there gap phases in the cell cycle? That's the G phase. What does G stand for? Grow. So cells grow, cells double their masses of proteins and organelles, They monitor the internal, external environment. So it's like a healing, growing phase. Beautiful, cool. Okay, cell cycle control. Okay, we're gonna get into it. We're also gonna get into pre-RCs and pre-ICs, because that seems to be tripping people up. But CDK, cyclin-dependent kinase. I love how it says it in the name because it tells you about how it works. So regulating it is possible through one, you can bind to cyclin, right? Cyclin in different phases. There's specific cyclins in different phases. CDK, it's cyclin dependent. So it needs to be bound to cyclin in order to be activated, right? So that's a way we can regulate it. Another way, kinases and phosphatases. CDK has to be phosphorylated by the CDK activating kinase, CAC. Another fun name that tells us a lot about it. WEE1 kinase inactivates CDC via phosphorylation. Phosphorylation must be removed. The term, this is kind of confusing, but the term phosphorylation means like removing, but I don't know why that word specifically there, but don't get confused. So, so obviously it's bound to cyclin, activates, it's bound to cyclin, cyclin's gaining, and then it is phosphorylated by that CAC, which is the cyclin, the CDK activating kinase. And then WEE1 removes that. Okay. And there's CDK inhibitors. So it binds at the active site near and inhibits ATP and substrate binding. We'll talk about that. So different cyclins help CDK target different substrates, right? Different cyclins for different parts of the cycle. So when CDK needs to make specific interactions, that cyclin is important because that specific cyclin from that specific phase is going to tell the CDK what to do. Straightforward. And the cyclin itself is regulated by production and destruction. So the levels are going to rise and fall. And so if there's ever a question that's like, okay, we want to change what CDK is doing, we'll talk about pre-RCs and pre-ICs. A lot of that is dependent on the cyclin that we have. So does anyone remember ubiquitination? Anyone remember what that means? It's like when it gets like tagged basically to be sent to the proteasome. Proteasome is like the garbage disposal of the cell. So you get tagged with that cyclin and then it's going to be destroyed, which means CDK is not going to be active. Okay. Okay. So name one way that the CDK activity is regulated and briefly describe how it works. Somebody just give me one. We'll just go through this pretty quickly because I kind of just went over it. Yeah. Cyclin available or unavailable to bind to CDK, that can regulate it. CDK degradation, if you don't even have it, can't do anything with it. CDK inhibitor, in the phosphorylation, dephosphorylation of CDK. So that's that WEE1 kind of that's going on. So that's that WEE1, and that's the CAC, which is, what does CAC stand for? CDK activating kinase. Okay, so checkpoints. They are the brakes. Okay, something's wrong. We want to check it. We want to keep it going. So they, a lot of things can happen. So is it has enough nutrients in the G phase? Is there DNA damage? Is the replication even done yet? We're ready to go on to the next phase. And is there proper spindle assembly, which we associate with what? Microtubules. Okay, at which checkpoint? There's the spindle assembly checkpoint, right? And there's also the G2. Okay. If cell pauses too long, cell's gonna die. It's too long, nothing's happening, we're just gonna die. P53, she loves P53 and P21, okay? P53, central regulator of stress and TF. So it's constantly made, it's constantly degraded, and it can serve as an upregulator. The whole point of this guy is to upregulate P21, which is a CDK inhibitor. Okay, that's this whole guy's job is linked to P21. If we have a bunch of this, then it wants to get more of this. P21, which is CDK inhibitor, to inhibit the cell cycle from continuing. CDK inhibitor, it's going to inhibit things. Cool. Okay, one cyclin binds CDK. Is CDK fully active or no? No. Why? Yeah, you said it too. CAC, right? So it's not activated. It's getting there. We're on our way. But we need CAC, the cyclin, the CDK activating kinase, to phosphorylate it for it to be fully active. Kind of like what we were talking about before with like AKT. You can have stuff that helps it activate, but there's always one thing that needs it to completely activate. And in this case, it is the CAC, the CDK activating kinase. What takes the phosphate off? What is that guy called? Yes, what's the name of that guy? What's the name of that guy who takes off the phosphate from CDK? We won. Right? Remember that. Oh, so here, let's go back. Oh, yeah, no, you're right. Hold on. G1.
I thought we wanted to spot correlated with like different areas.
Yeah, sorry, I'm wrong. So there's the one phosphate, the phosphate that activates it, adds again, inactivates. Okay, is that clear? Sorry, that's my fault. We1 is a kinase. Kinases add phosphates. Okay, cool. Okay, checkpoints. So it's a good photo of all the checkpoints. So we have that G21, we have the M, cool, important G1 events. This is like synthesis of like everything, like summary of everything that we're about to talk about in case that you don't remember this. So growth factor comes in, goes through the RAS MAPK pathway. Cyclin D is made, okay? So cyclin D is binding to cyclin CDK. Are we fully active yet? No, right? It's helping, we're on our way. We're not there yet. So the cyclin D phosphorylates RB. RB, these guys, it's really similar to BAD. Okay, so when things are activated, it's going to phosphorylate RB, and RB is going to let go of E2F. So RB before, RB after. And it needs that cyclin CDK to bind before it can do that. Okay, it lets go. When E2F lets go of RB, it's just E2F is now fully active because it's free. Ooh, it's free. And then now it can go and do its thing. So now it's going to go and say, all right, we're good. Keep going. That's the restriction point. So it's just like BAD and BCL2. We let go. When we let go is when we can actually do the thing. Oh yeah, and it's a positive loop, meaning that a downstream regulator is causing regulation of upstream in the same pathway. Okay, so before committing to the cell cycle, cells need to check for enough nutrients, right? No DNA damage, and need to grow. Is it grown enough? So for this reason, E2F is bound by RB until the cell is ready to progress, upon which G1 cyclin CDK activates, phosphorylates RB. Phosphorylation of RB leading to activation of E2F is the point of no return. This is just a fancy way of saying that until this is phosphorylated, we still haven't committed yet. When this is phosphorylated, we've committed because then E2F is gone. Now we're committed. We're in the cell cycle. Okay. So what happens if these two stay together? Are they going to keep going? Is it going to start replicating and stuff now? No, it's not. So it needs to let go before anything can happen. So let's talk about pre-RCs. So pre-RCs form when CDK activity is low in early G1. So again, this is like the building action, right? So CDK activity is low in early G1. It's called origin licensing. She ever asked you about it? I don't think she will. But if CDK activity is high, or if it's active, this complex is not going to form. So we're used to seeing things that's like, oh, if things are active, things are going to happen. In this case, CDK activity needs to be low in order for this to form. So ORC binds to the origin of replication on the DNA. ORC recruits this guy, and then that guy recruits all of these guys. They're just fancy names. Now, ATPase activity of CDC6, this guy, allows MCM, these two guys, to be loaded onto it. The MCM complex is helicase. And helicase is the enzyme that unzips DNA. It's just a zipper. That's all that enzyme is. And then finally, these guys leave the origin. So IC, so this is pre-IC now. So pre-RC comes first, pre-IC comes after. So before CDK activity, is it low or high? Before this? Low, right? Low. But things need to be low for this for the pre-RC to form. Now we're at pre-IC. Pre-IC, now G1 CDK is active. When we talk about G1, we're talking about G1 cyclin, right? That's what that means. G1 CDK is active, more proteins are going to bind at this complex that we just made, creating that pre-IC. So pre-RC comes first, things add to it, it makes the pre-IC. DNA polymerases are loaded onto the origin replication. Replication begins. Do you remember what DNA polymerases do? They just recruit, so they just like start transcription essentially. It's called origin firing. It's going to load onto here, and replication is going to begin. And not all origins fire at the same time during S phase. Okay, summary. Where is the point of no return? Someone tell me. What's the point of no return? What's going to happen? Super quick. Yep, so E2F is going to let go of RB because RB got phosphorylated. Now we've committed. Now we're officially in it. Point of no return. G1 early, CD activity is high or low? Low, letting us form the pre-RCs. Now CDK and the G1 cyclin have bound together. Doesn't mean it's active. It's just, they're just together. The CDK inhibitor, which is WEE1, which adds a phosphate to turn off CDK, binds and inactivates till it's ready. And then when it's ready, CDK is active, is the removal of WEE1. Yeah, we already said that. Releasing E2F. Oh yeah, and E2F is a transcription factor. Transcription factors tell the DNA polymerase where to go to start transcription. Let's tumor suppressor. Okay, I think this is a good question. Okay, is E2F a tumor suppressor or an oncogene? Why? So if E2F is activated, if it lets go of RB, what's it going to tell the cell to do? To go into S phase. And what happens in S phase? DNA replication. So would we call this a tumor suppressor, which means it's preventing cells from like replicating and doing that thing, or is it encouraging it? Is it the proto-oncogene? That's what we call it. Yeah, so when it lets go, more replication is happening, more cells are being made. Okay, RB, tumor suppressor or oncogene? Suppressor. Why? Right. So if it's here, we'll just, yeah. So suppressor, because it is, if it's not, phosphorylated, it's going to keep holding on, which means we're not going to get into cell replication. WEE1 adds the inhibitory phosphate, meaning that it's suppressing. Okay, because it's turning off CDK. CDK inhibitors, suppressors, P53. Remember, P53 upregulates what? What's the other P? P21. P21. Does anyone remember what P21 does? Somebody say that louder. Yeah. So it's like, bad things are happening. And then CAC. CAC is, what does it stand for? Yep, CDK activating kinase. So it activates things, right? Letting it go into DNA replication, meaning it's an oncogene. Okay, that's it. Sorry we kept you guys so long, but I hope it helped a little bit. We'll send out the recording if it saves properly.
Yeah.
What?
Yeah.